Thank you. My name is Brandon. Like she said, I'm a postdoc with Katie Lauderhouse at Northeastern University. And this figure on the right is from climate data that I downloaded that expresses climate novelty in North America. So these lighter areas of the map represent geographic areas that will have no climate analog to previous climates in the past. Honestly, and with so much of that map having that light color, it really stresses the importance of having solutions for adaptation and conservation and restoration. So as an example of California, we often think about trees as being locally adapted to climate, but in some cases they lag behind climate. And so Victoria Stork's lab published a paper not too long ago that compared temperature differences between the home environment and the common garden environment, and then measured relative growth between those transplants, and found that you get higher growth when you plant in colder environments, suggesting that these trees, these California valley oaks, are adapted to previous climates. And forest productivity writ large, so valley oak and all the other species, if we're not implementing climate-aware intervention, we'll see severe declines in forest productivity, which means forest fires and disease and everything that comes along with it. And so obviously these common garden and transplant experiments are resource and time intensive, and so there's been a push to kind of circumvent that time lag with using genomic and climate data to predict maladaptation to future climate. And so what goes into these genomic offset models that predict this maladaptation is climate data and genetic data. And once you put that in, you get this model that should give you some idea of fitness. And in this example, we have two populations that occupy different spaces of the climate space. And given their genetic constitution, they lie on that fitness line. But for the same degree of climate change, we might expect that these two populations might be more or less adapted to that future climate. So that population in B, that genetic constitution doesn't have to change very much to keep up with that genotype-climate relationship, whereas in population A that does. So the population A would be expected to be more maladapted to future climate, and it would show less productivity, growth, and all of the other things that go along with it. And so these genomic offset methods are used to sometimes predict maladaptation to future climates, but they can also be used for a specific restoration site. Let's say you want to plant seeds in a restoration site, you could rank source environments and predict how well those environments will do in that environment. And that could be within the range for assisted gene flow or outside of the range for assisted migration. 2 years ago at the EvolTree Conference, I presented an empirical study where I employed these two methods across two varieties of Douglas fir. So the interior variety of Douglas fir here in purple, this is these dots up here, and the coastal variety of Douglas fir here. And we see this negative relationship that we expect between some kind of fitness proxy and genomic offset, where high genomic offset means low growth or low fitness across those populations. But that was when we validated it within a common garden in Vancouver. When I projected that climate, those models to future climates, I got two different answers depending on which populations I used. On the right-hand model, I used both varieties, and in the area in circle said that that geographic area was most maladapted. And on the left, I only used interior populations, and then it said that same area was the least maladapted to future climate. So it really highlights the fact that we really don't know when these offset methods are going to work, and we don't know when they're going to fail. And really what that comes down to is understanding the domain of applicability of these methods, which is the circumstances or factors which a method is acceptably accurate. The schematic on the right or on the left kind of shows that where you have different factors which may or may not lead to an accurate offset model. So within the context of genomic offset, those factors are things like evolutionary history or experimental design that actually goes into the training and validation of the model itself. So if we want to have a better idea of this domain of applicability, we can use simulations where there's no error. We know fitness, we know climate, we know genotype without error. And that can really help us understand when these methods will work well and when they won't. And so simulations that capture biological realism, contrasting cases, and adversarial scenarios are really what we're going for. And what I mean by that is by biological realism, so we can think of things like environmental climes. Here is surface temperature of the oceans. Katie is a marine biologist, so that's where that comes in from. Contrasting cases where you can control for certain aspects of the simulations while allowing other aspects to vary. So you could vary population connectivity, having island, isolation by distance, or expansion, but controlling for differentiation among your populations. And adversarial scenarios means parts of the data that capture extreme characteristics of the empirical data that we're really trying to learn about, such as these novel climate scenarios in the future. So these next couple slides, I'm going to be going over those simulations that were developed, and this all comes from Katie's 2023 PNAS paper, if you've seen it. The first landscape has two environments on it. One is the temperature environment, and the second is just a generic environmental factor. The temperature goes in a north-south gradient, and then the second environment goes from the east to west gradient. A second landscape is what we call the mountain landscape, and you can kind of just see this kind of ridge line type scenario in this situation, where you can kind of think of this as an elevation type landscape, where you have similar environments on either side of this ridgeline. And because we're also interested in marine systems, we have this estuary type scenario where on the far right you have connectivity and gene flow in the ocean areas, but as you go into inlets, you can decrease in salinity from the ocean environment to these river mouths. And so notice in this estuary environment, there's no connectivity, there's no connection between these populations, between these inlets. One of the things that I want to also point out is in these two environments on the left and the right, geographic distance corresponds to environmental distance. But in this middle scenario, those two environments on the top and right are the same, but when we put populations on a landscape, we don't expect their genetics to be the same either. So will this challenge models for offset, or will we still get a decent result? These simulations always also varied the level of pleiotropy. So in one case, we just had a simple adaptation to a single environment, which is temperature. In the case where there was two adaptive traits, one was adapting to temperature and one was adapting to that second environmental variable with no pleiotropy. And then in this other situation, we allowed the alleles to have effects on one or both traits. And on top of that, we have these traits either experiencing equal levels of selection intensity or differing levels of selection intensity. We also varied 5 demographies. So on the left is the most simple one where we have constant population size and constant migration and symmetric migration between all populations that are connected. The second one represents some breaks where we have some declines in that connectivity, so some kind of vicariance event. In this middle one, we have a north-south cline. Maybe these population sizes will also lead models of stray because of drift and allele frequencies differing from the genotype climate pattern that we expect from adaptation. Another situation is pretty common where you see larger population size in the center of the range where that declines to the edge. And one, we're just kind of mixing everything up. We have variable population size and variable migration rates, and some of these are not symmetric. On top of these, we have 3 genic levels. We have an oligogenic, a moderately polygenic, and a highly polygenic model where the underlying architecture is either underlain by a handful of loci or many, many loci. And if that wasn't enough, we had an additional scenario where we simulated a complex multivariate case. In this case, there's 6 environmental traits that are under selection. They're moderately polygenic. There's no limit to pleiotropy. So when alleles become adaptive, they can affect 1 trait, or they could affect 6, or as many in between. There's also this added layer of range expansion from multiple refugia. You can see that with these ancestral clusters here. And we used real climate data and put it onto a landscape and then simulated evolution on that landscape. And these are just temperatures that we took from British Columbia for mean annual temperature, driest quarter. And so these climate variables here are relatively uncorrelated. And so we wanted to test genomic offset methods, and that just didn't mean one method, that meant as many as we could within reason. And so obviously we tested gradient forest, which is one of the more popular ones. This one can accept multiple environments when you train the model, but it doesn't apply any structure correction. RDA is another one, another offset method that's been proposed. It doesn't do structure correction by default, but you can impose structure correction if you would like. It can handle multiple environmental variables at the same time. This LFMM2, which is the genetic offset, not the genetic gap, out in the LEA package, is multivariate, and by default applies a structure correction. And this last one was the risk of non-adaptiveness. It is not multivariate, so each offset is a single environmental variable at a time, and there's no structure correction. So into these offset methods, I fed them allele frequency data, and I varied the type of marker set data that I gave to these offset methods. Either I gave them the whole data set of markers, or I gave them adaptive alleles, or the ones that we knew were strictly neutral that were on chromosomes that didn't have any adaptation on them. I won't be talking about the neutral loci today, and I'll save that for the publication. And so, so most of these models that I'm going to talk about today, I only gave them in the adaptive environment. So for simulations that had one trait, one adaptive environment, I only give them that environment. And when they had 2 adaptive environments or 6, I gave them those environments, no more, no less. So within each of these landscapes, I was interested in how well these offset methods were variable across the landscape. And so what I did is, within each landscape, within each level of these methods, is I did a full reciprocal transplant. This is just showing like a bottom corner of one. But I projected offset to all 100 populations' home environments, and I evaluated the performance as a rank correlation between offset and fitness, where we expect, like I showed you with the empirical data, that we should see a negative relationship between those 2 measures, where when offset is high, we should expect fitness to be low, and when offset is low, we expect fitness to be high. So in total, there's 3.9 million 1 and 2 trait within landscape offset evaluations. I was telling other folks the other day, it took 60 years on the computer to do all of this. And so we have levels of these represent the evolutionary history from simulations, these offset methods, the marker sets, and the common gardens. And so the part of the domain of applicability is this evolutionary history, which comes from the simulation parameters, and the experimental design, which is not only marker set, but I forgot to put on climate data here. So the things that you put into the model and how and when and where you validate it. So the first question I had was, how do those simulation parameters affect performance of offset methods? And the second question was, does it matter which markers I use? Does it matter if I give it the whole data set, or what happens if I just give it adaptive markers? And the third question I had is, how well do these methods extrapolate to novel environments outside of the range of environments used in training? In a lot of the machine learning literature, we see that if the test environment or the test data differs substantially from the training data, we're not going to see good predictions. And so a lot of these methods are projected 100 years into the future. Can we do that? And the last question I'll address is, how does the addition of non-adaptive nuisance environments in training affect performance? So I told you earlier that I only gave these methods the adaptive environments. What happens if I give it additional environments? So often, especially in terrestrial systems, we have a large number of environmental variables that we can feed these models. What happens if we give it that has nothing to do with adaptation? Will that matter, or will it decrease performance? So a lot of these figures will have this y-axis where I measure performance. I have the negative values up top just because it makes it more intuitive. So these are better performing models, and things down here are worse performing models. And so I did an ANOVA with all these simulation factors, and I found that local adaptation was one of the highest degrees, or one of the main factors that predicted this. But even so, these are the 3 landscapes that I showed, and in each of these landscapes, as local adaptation increases, so does model performance. But you can see there's not a tight linear relationship, so there's caveats, there's things I can't talk about today. I'm happy to answer questions as best I can. But that said, still, local adaptation within the populations that go into training matters. And so this figure I did with only 2 trait simulations, and I only evaluated with all markers. And so each of these dots is a median value from each of those 100 common gardens. In this figure, I have all common gardens represent. And so I have RONA, the 2 environments for RONA on the left, LFMM, RDA without structure correction, RDA with structure correction, and then gradient forests. And the first thing I just want you to see is that most of these methods do better in that stepping stone landscape where you just that first landscape that I showed you, where It was just a simple Klein environment. And that mountain landscape, which was the middle landscape that I showed you, where I had those two environments where I said the genetics was going to be different, that decreased performance. I think it led the models astray. And then in that estuary Klein's situation, we had the worst methods of all, except for this RDA corrected. Within the simulations, Rona, as you can see, had the highest variance in prediction ability across the landscape. So I'm going to leave these out for most of the graphs. But I want to leave the caveat is with the empirical data, I found similar relationships between Rona and gradient forests. It's going to be a common theme that there's a lot of nuances with this. So I'm going to be showing you mostly these figures. I'm going to also exclude the structure-corrected RDA from most of these future figures. So the first part of this, we conclude that local adaptation is a primary influence on some landscapes, and some landscapes do reduce performance. And so does it matter which genetic markers are used? So this is a figure that I show a performance of models that have only adaptive loci versus those that have the whole data set. And up here is where we want the models to show up, and these are all negative values. And this is the one-to-one line. So anything, if you see any skew on this side, that means the adaptive markers are providing some benefit. If you see any skew on this side, that means the whole data set is providing some benefit. For LFFM, there's only some slight benefit in some situations for adaptive markers. But when we move up to gradient forests, we see more of that effect. We see more of the skew on this right-hand side that indicates that there's adaptive markers are important in some situations. We can also see that in some situations, the whole data set did better. In RDA, the uncorrected RDA, we see even a higher proportion. This figure doesn't illustrate it so well, but in these low-performing models, those are usually the models that have low local adaptation. And so what this also says is that those adaptive markers are more important when local adaptation is low. And so this is the sixth trait model now, and so we only had one replicate, so it's just the Hunter Garden data. It's not sorry, I also mentioned that this is a scatter plot that shows you the density of markers to kind of give you some idea. So most of these are on the one-to-one line, but some of them are quite off. And so this is not a 2D scatter plot. This is just the results from the 100 carbon gardens. Some methods are quite variable when you give them adaptive markers versus all, like the RDA structure correction. But gradient forests and this RDA uncorrected are less variable. So adaptive markers provide varied advantages across these scenarios, and are particularly important when local adaptation is low. And so how well do they extrapolate to novel environments? So we can think of some climate gradient where we have populations that we've put into our training data and some new environment that we haven't sampled that we'd like to know performance. So we have some fitness function that is real that we haven't measured, which could differ in those new environments. And we don't know which one of these could take place, and this could be due to biological reasons. Like if you plant a palm tree in Alaska, it's not going to grow well. That's one of the C scenario, but for any other reason, it could be any of these scenarios. But in our models, A is the true scenario. Fitness just follows the same relationship no matter what environment we put it in. So can these models still predict that, ignoring these other biological scenarios? And so what I did is I created new environments that are increasingly far from the training environments. So this is just a PCA data of the environmental data. All these black dots are the population climates that went into training. The blue ones are the novel environments. And the PC1 and 2 and PC3 and 1 look the same, but they're standard deviations from the center of those climate variables. And so those blue dots are here on this x-axis, and over here, I'm just going to give you some kind of somewhere to kind of understand how they compare to the data within the landscape. And I think what you can see is immediately is that these red populations that have high local adaptation fall off faster than the populations that have lower local adaptation. So before I told you local adaptation was important for model performance. But these are also the same populations that we won't be able to predict in new environments, at least in these simple simulations. These are another of those heat maps. Before I showed you that there was variable performance or variable advantage to using adaptive markers, but particularly for gradient forests and the RDA, you can see this large skew on the right hand that indicates that adaptive markers are a lot more important in these scenarios when you predict adaptation to novel environments than when you predict adaptation to environments within the landscape that you've used in training. And so this is just another figure that's illustrative of the fact that within the landscape performance is non-indicative of how these models will extrapolate to new environments. So we can go out and use a common garden to validate these things, but we still don't know if it's going to predict well in a novel environment. So I've lined this 6 trait figure up with the 0 and the negative 1 up here, and what I want you to see is these methods do very poorly, and LFMM is basically telling us nothing. So the performance declines with climate novelty, and adaptive markers provide some advantages within landscapes, but more advantages when you project them to novel climates. But again, there's caveats, and that's not always. And so the last question I'll address is, how does the addition of non-adaptive environments affect training performance? So this first figure is going to show you the 1 trait single environment scenarios. Everything I've showed you up until this point was this first dot, where I've had no other nuisance environments. So all the environments that I've put into training were adaptive. And these other dots, these other colors of green, are the addition of 1, 3, or 4 additional environments that are uninformative with regard to local adaptation. And for all of these methods, these performance declines as you add environments that don't matter, particularly with RDA, less so with gradient forest, but there's still quite a big fall in performance. And we see similar things with the 2 trait scenarios, not to the same degree. And with gradient forest, it somehow is stabilizing out here. And I think that's just one of these situations where gradient forest, this is just a matter of this situation, this 2 trait situation. And the same thing with the 6 trait scenario. We see model performance decline when we add environments that aren't important to local adaptation. So in each of these programs, we see that the ones that are trained with adaptive environments versus ones that are trained with non-adaptive environments reduce performance. So the addition of non-adaptive environments decreases performance. And so where I see these offset methods going is everything I presented to you today was pretty simple scenarios, 1 or 2 adaptive environments. I think there's a somewhat realistic when I use empirical data. Gradient forest will kind of tell me which climate variables are most important to predicting the layout of allele frequencies across the landscape. And usually it was 2, 3 that really stood out from the rest. But still, even beyond this, we still need more evaluation with empirical data and in simulation studies. And what I mean by that is when we employ these offset methods, we should be varying things that go into the training data to see how it affects the outcome at the end. So marker set, just like I had the whole data set versus things that I thought were adaptive versus neutral. You won't have neutral markers in empirical data because of confounding effects of other things that are going on the chromosome. But you could also think of using stricter filtering of either these outlier sets that we associate with climate, or you could think about at the earliest stages when you're calling genotypes or estimating allele frequencies, only using those genotypes that have really high quality scores. I know when I first started my PhD, I really had trouble throwing away SNPs. I had really large trouble throwing away data, but in these situations, I think those are going to matter all the more if we have really good estimates of genotype or allele frequencies going into these models. So another thing that we can vary is the climate data that we put into these models. Often we're not going to know which climate data is going to be important. But we can vary those climate data. So what I mean by that is, in my empirical data, I used climate normals from 1960 to 1990. What I could have done is done sliding windows across those decades to see how that affected my performance, because we're assuming that much of the adaptation is due to pre-anthropogenic warming. That's why we use 1960 to 1990. But that might not always be the case. What also might not be the case is that you have different populations in the landscape that are adapted to different environments or different time periods too. And also varying the set of populations that you use for either training or validation. In the empirical study, I showed that my inference on how well the model performed depended on which populations I used to validate it. But also I showed that when you vary the populations that go into training, you can get a lot different answers in the first version of our publication. So I think populations that go into training should also be varied as well. I think it should be particularly important to pay attention to these populations that are isolated or these, that experience structure that's a lot different. So yesterday we saw Jill's presentation where she did a GEA and she said that the answer differed when she included Norway or when she excluded it. And that's exactly what I'm talking about here. If we include populations that are highly divergent, I think that's going to skew models. And with our empirical data, the coastal variety of Douglas fir had that highest connectivity. So when I looked at it on a PCA, it was just a smear. And so there was very little population outliers like that, but there were still some that we could have excluded had we wanted to make a more complicated paper. If we have no idea of which environments are going to be adaptive, the ones that are temporally autocorrelated are probably a good bet. Consistent environments give opportunity for local adaptation, and we know that from the literature. But also spatially autocorrelated environmental data will also kind of provide that same benefit. So if we have no idea what to use, we can start with these type of data. And I was just thinking about this the other day. If we want to vary the environments that go into training data, we can pick maybe 5, and we can do a factorial treatment. 5 factorial is 120, so you can do 120 models to see how that answer varies. And like I said, so many dozens of models are going to need to be evaluated if we want to incorporate these types of genomic offsets into forest management, especially if we want to think about mitigating uncertainty and things like that. And that doesn't matter how well your sample design is, we're going to have to evaluate these models. Again, a good within landscape validation from a common garden that we use today does not necessarily mean that we're going to have a good prediction in the future. That's just something we need to be aware about. And we also need to be aware about variation in local adaptation among training populations. In the simulations, we had very little variation in local adaptation among the populations, but I still saw a minor effect of this, where the more variable the local adaptation is in the training populations, the less the models performed well. And so because of all of this, genomic offset methods might not be good for assisted migration, and they might not be good for predicting maladaptation to future climates. But given that we can, there's a few other studies empirically that show that we can validate these models within a landscape and the results from these simulations, maybe these offset methods are better suited for near-term assisted migration. One other thing I didn't mention is some of these methods don't predict the same across the entire landscape. So you might have a good validation where you want to apply it somewhere else on the landscape, and your model could be totally wrong. Of course, there's many caveats. These are simplistic scenarios. I made a list that I want to get to. They're not universally applicable. So I think if we're going to have, we can tailor our simulations to our empirical data. We can also vary the measure of performance. So I did a correlation between the predicted offset and the known fitness of all populations. But maybe what we're interested in is not how well we can predict all of those populations. Maybe what we're interested in are the populations that are least adapted or most adapted to a particular environment enriched in the tails of that prediction. And so maybe we don't want to use a correlation statistic. Maybe we want to use some kind of enrichment type analysis. I'll put away the list. Also, although RONA didn't perform well in these simulations, like I said, it did fairly well in empirical situations. And this is also the most simplistic version of RONA that's been proposed. There's other versions of RONA, such as weighted, one that weights a loci differently. There's also ways that we can extend RONA to a multivariate type offset method. But I think the first step is understanding how the basic RONA model works in these scenarios, and then we can add these complex layers onto these methods. And another thing about gradient forests is we very infrequently do hyperparameter tuning. And what I mean by that is these random forests, you give the model, or you give the program tree depth, you can give it the number of nodes on a leaf, all these different things that can perfect the performance of a random forest, and we rarely vary those at all. So we should also think about how those hyperparameters of these models will affect the outcome in the future. One of the things I've been thinking about as I heard other people's presentation is we're using these single locus GEAs, but recently for the co-adaptive project, which I was a part of, but I wasn't a part of these papers, Tom Booker, Sam Yeaman, Mike Whitlock, and others came up with a method that uses a window approach. We can avoid that problem that we always come to with structure correction, where we can look at the signal within these windows, and then what this first method does is it leverages the level of linkage disequilibrium that we would expect from local adaptation. And by not correcting for structure, but taking advantage of that LD, we can have an expectation of whether this genomic window, perhaps a gene, perhaps a sliding window, is more indicative of a region underlying local adaptation. And for those of us that have really close related species where we're looking for adaptive regions across these species, they also developed a method to look at a convergence. So I think as long as we have orthologs, we can test this idea, is there convergence? And if there is, I think that's added support that these regions that we're thinking are adaptive truly are. I want to thank you all. I want to thank the organizers for the invitation. Of course, my advisor, Katie Lauderhouse. I just updated the bioarchive of our empirical offset paper that I showed you earlier. It's on bioarchive right now, and that's the bit.ly link and the QR code for that paper. So if there's time, I can take some questions. The first talk without questions.
Oh,
thank you.
First question.
So, I mean, the beauty first of the simulation is you can do anything, and you never stop. So that's not what I'm asking you here. I'm trying to understand what you did. Is it true that the covariance structure of the climate is not changing? It's just maybe sampling that PCA space? So I mean, you're not truly simulating changing covariance structure of climatic variables through time. The model's actually not even worse than this.
Right, so I don't think I measured the covariance among those environmental variables, but the distribution of those environmental values are the same across those landscapes.
Yeah, but the covariance is already in that PCA offloading version. So if you're sampling, were you sampling from inside that original PCA space?
Oh, so what I did is to get those novel environments, is that what you're talking about?
Yeah.
So what I did is I calculated z-scores I used the training populations, those black dots from those PCAs to get a mean and a standard deviation, and then I just went outward using a z-score.
Okay, so it's kind of in there, but you're sampling novel parts of the space.
Yes, exactly. That's
okay,
what we were
cool.
trying to do.
Thanks.
Santi?
I wanted to ask, like, the genomic offset methods is basically static in the sense that you base your predictions in the environment gene correlations that are currently in the landscape. So have you tried to include, like, the potential for short-term adaptation in the simulations? The population may change when there is a new climate.
So if I understand the question correctly, you're asking if after I train the model, if the environment changes?
Well, no, like you have a population, right? It's going to be a change in climate. and then the full allele frequency climate correlation may change.
Yes.
So then the prediction will be based in a model that is not currently adequate to. So.
Yeah. So one of the assumptions of these genomic offset models is that the time that you sample and the time that you're trying to predict, nothing's going to change about the relationship between climate and local adaptation. But of course, it probably could, because you could have situations where populations like have gene flow into them, they receive adaptive alleles, and then the future outcome is different than what we expected. And so that's another reason why I suggested near-term assisted migration, is because that's less time for those type of situations to occur. Good point, son. Thank you.
Hi, Brandon. Really interesting talk. Thank you. Could you say a bit more about what you were saying about using enrichment analysis in the tails?
The way people have done this is they created a model that relates offset and some measure of fitness. Empirical data, that's some proxy, and simulation data, that's real fitness. So that's either a linear model or a quadratic model. Some R-squared value tells us if it's good, or some correlation statistic tells us if it's good. But maybe we don't need the model to predict the worst populations. Maybe we want it to predict the best populations, and that's maybe all we care about. We don't need it to predict everything. We just need it to predict the best or the worst. And so if there's enrichment for these, so in simulations, since we know what's going to perform the best and what's going to perform the worst, we can do some kind of enrichment tests for those genomic offset predictions. Do they predict the real populations in that top tail of that distribution or in the bottom tail of that distribution? So that's what I mean by enrichment.
Thank you, Brandon. Very extensive investigations as always. I just wanted to add another assumption to the whole thing, which makes it probably even more critical. In the simulations, you simulate locally adapted populations, I guess, and you try to evaluate genomic offset. But in natural populations, we assume that these populations are locally adapted, and we know this is not always the case. So this actually makes the prediction of the genomic offset prediction even more complicated, I think.
Yeah, exactly. If you're trying to create a model between climate and fitness, and the data that you're putting in don't reflect a relationship between climate and fitness, that prediction is probably going to be wrong.
Yep.
Thank you for your talk. I have few questions. Just correct me if I'm wrong. Did you consider those traits, 2 traits that you have simulated, are they positively correlated, or would it be possible to consider them as negatively correlated traits?
Yeah, so I think the next step is doing things like where we have antagonistic traits. Because of the way that those traits were laid out on the landscape, I don't think there was a strong correlation between them. And there definitely wasn't for the environments that I added as nuisance environments. There's very little correlation structure among those environments.
The second question is, would it be possible to develop a precision modeling for each environment or each landscape exclusively, instead of considering a common model for the whole landscape system? Could it be possible to develop a model for the exclusive landscapes that you are discussing here?
You mean like a simulation model?
Yeah.
Yeah, I think so. I think so. People, when they want to understand the power of a GEM method, they can simulate their data that reflects the true parameters of the populations that they're sampling, and to understand how likely that they are to detect different alleles of various effects. And the same thing goes with the simulations that you have, that you want to test ideas about your populations and how well they might predict in different environments.
How significant differences that you see when you equally weighted or unequally weighted traits?
You mean when I showed that variability in the selection intensity?
Yep.
It's still up there. So really all that did was, I can't go back. Well, really all that did was affect the level of local adaptation. So let me try and go back. Can I get out of here? There we go. My computer's being slow, it's heating up. Okay. The very first figure. One of the first figures. So this. So on the right-hand side are the simulations, the yellow, the blue, and the green, are the simulations that had equal selection intensity. And on the left-hand side, the yellow, the blue, and the green, are the simulations that had unequal selection intensity. Oh, thanks.
Interesting. Thank you.
I think we've got time for maybe one more question. Okay, otherwise I wondered, you've actually put the slide before, you've got these three sort of scenarios, the clines, the mountain, and the estuary as well, which is inclines. And I wondered how well you think those represent most landscapes, because obviously the estuary is kind of a particular one, but actually you could possibly imagine it with, sort of barriers in the way anyway. And what impact do you think that would have?
I think the spatial autocorrelation of those environmental variables and how heterogeneous they are across the landscape would challenge models. And I think that is potentially a next step, and where you have those heterogeneous environments or pockets of particular variables. But when we think about these pockets of like a variable where it's high in one little pocket but low elsewhere and then high somewhere else, we can use those kind of mountain scenarios to think about how well we're going to be performing in that area. But definitely I think another next step could be simulating an environment where the Apache environment.
Brilliant. Thank you very much. I think we're dead on time, so that's perfect. Thank you very much,
Thank you.
Brandon.